So we are yet again going to continue the topic of digestion. In our previous class, we have seen digestion of carbohydrate. Today, we will see digestion of protein, its sources, how the digestion occurs, and how the absorption of protein takes place in the body. Okay, so as you can see on the screen here, we have sources of protein in our diet. So meat, egg, milk, nuts and legumes are rich sources of protein that supplies protein to our body. Okay, and wheat, okay, which contains glutenin and gliadin. Milk, the chief protein of milk is casein, also contains lactalbumin, albumin and myosin. Egg is also a rich source of protein and it supplies albumin and vitelin. Meat is a rich source of collagen, albumin and myosin. So, these are all some sources of protein to our body that we take through our diet. Next, how the digestion of protein takes place. Now, any protein that is formed of long chain of amino acids and they are joined by linkages known as peptide linkages. Okay, now these peptide linkages are similar to glycosidic linkages because they are formed by the condensation of a molecule of water, but the difference is that here it happens between a amino group and a carboxylic group. Okay, detail you might be studying in one of the papers. Anyway, recall a protein is formed of amino acids. Carbohydrates, their basic units are monosaccharides, and in protein it is the amino acids. So, these amino acids, okay, yeah, here you can see these are all different amino acids and they are present in the form of a chain, and these chain are folded in different forms to form the protein. So, that means any protein you take, it is basically formed of something called as amino acid. Okay. So, that means at the end of digestion you are going to get amino acids, the simple forms that can be easily absorbed by the body. But unlike carbohydrates, these proteins can also be absorbed not just only in the form of amino acids, they can be absorbed in dipeptide or tripeptide forms. Okay. Yeah. Next, moving to the next slide. So, digestion of protein, how does it takes place? Digestion occurs by the enzymatic activity, and again recalling, digestion can be of two types, mechanical and chemical digestion. So, here we are concentrating more on chemical digestion, and whenever the word chemical digestion comes, it occurs by the help of certain enzymes or the salts. Okay. So, here it is the enzyme, and the digestive enzymes are generally called as hydrolytic enzymes also. Okay. And here, since they digest the protein, they are called as proteolytic enzymes. Okay, they are called as proteolytic enzymes. Enzymes that digest protein are known as or is known as proteolytic enzyme. Now, where do you put your food first? You put it in the mouth, and we have seen that carbohydrate digestion starts in the mouth itself, but protein digestion does not occur in mouth. So, there is no digestion of protein in the mouth. Why? Because there are no proteolytic enzyme. Since there are no proteolytic enzyme, no digestion in the mouth. Okay. That means the food that is digested mechanically in the mouth, which forms a some soft ball, will be swallowed in the form of a bolus, and this swallowing is called deglutation, goes through your esophagus and reaches the stomach. So, when it reaches the stomach, here the actual digestion of protein begins. Now, let us see how the digestion occurs. So, in stomach there is a enzyme called as pepsin. Okay, and this pepsin enzyme carries out the digestion of protein. There is also another enzyme called as renin that is found only in case of infants, that helps in digestion of milk, but in adults we have only pepsin that helps in digestion. Okay, this pepsin, it is secreted, okay, as a pepsinogen. This pepsinogen is an inactive form of enzyme. So, unless there is food in the stomach, Okay, this pepsin will be present in the form of pepsinogen. Pepsinogen is the inactive form of enzyme pepsin. Okay, and this is secreted by special type of cells in the stomach called as chief cells. Okay, and this pepsinogen, it is activated by gastric hydrogen ion, and that is nothing but, the source of this is nothing but hydrochloric acid. We all know that stomach secretes hydrochloric acid that actually gives a pH of around 1.2 to 2.2, the gastric juice, and this at this acidic pH, pepsinogen is activated to pepsin. Okay, and the pepsin, this particular enzyme pepsin, can act optimally at a pH of 1 to 3. Okay, at a highly acidic pH, this enzyme is active. pH you must be familiar again. This is the a measure of active hydrogen ions that is present in a solution, negative logarithm of base 10. But anyway, we do not want to go for mathematical expression. So, remember, a pH refers to the amount of active hydrogen ions present in the sample or a solution. So, more the hydrogen ion present, more acidic the sample is, less the hydrogen ion present, less acidic the sample or solution is. So, that means when you say pH is 1 to 3, it is highly acidic, because pH range varies from 1 to 7. 7 is neutral. So, anywhere between 1 to 7 is acidic pH. So, here 1 to 3 acidic pH means it is highly acidic pH, and pepsin is highly active at this particular pH. Now, this pepsin can hydrolyze the proteins that are present into peptones and proteoses. So, as soon as the pH increases above 5, the activity of this particular enzyme stops. So, this is how the activity of pepsin is regulated. Okay. Now, I am going to the next slide. So, here what happens, let us see how exactly the pepsin is helping in digestion of a protein in the stomach. Now, there are different types of cells present in the stomach, and they secrete different chemicals. Some secrete mucus, some secrete hydrochloric acid, some secrete these enzymes, and there are also hormone secreting cells. Okay, we are much bothered about the enzymes that are secreted. Now, what happens? If you recall, digestion is of two types, mechanical and chemical. Here, mechanical digestion of the food takes place by the muscular contractions, okay, of the stomach. And chemical digestion takes place by two ways. One is by the acid, the hydrochloric acid that is present, and the other one by the help of the enzyme pepsin present in the gastric juices. Now, here you see, I said pepsinogen is secreted in its inactive form. Unless there is a food, this is going to be in the inactive form only. And then what happens next? When the food comes to the stomach, hydrochloric acid is secreted by the oxyntic cells present in the gastric gland. Now, this hydrochloric acid has different function. It kills the bacteria, it softens the fiber, etc. In addition to that, it has a major function, that is activating this pepsinogen to pepsin. Now, what is this activation? Activation refers to a condition where usually when an enzyme is secreted as it is, particularly for a proteolytic enzyme, to protect from its adverse activities, these enzymes will have additional chains, caps and the tails will be present, or they will be folded in a different way. As soon as there is a necessity of activity of the enzyme, okay, some factors will help in removing those additional caps and the tails, so that the enzyme is now ready to act. This is to protect the rest of the normal parts of the body or organ or tissue from the activity of particular proteolytic enzyme. Okay. So, pepsinogen is in the inactive form. As soon as food comes there, due to the secretion of hydrochloric acid, pepsinogen gets activated into its active form called pepsin. Now this pepsin itself can activate. So autocatalysis can also take place. So that means once of some amount of pepsin are produced, okay, they are going to activate the rest of the pepsinogen. That is one way. There is another view that it says that, so in the presence of hydrochloric acid, okay, in an acidic condition, pepsin can also activate the pepsinogen to pepsin. Okay, whatever pepsinogen is activated to pepsin in presence of hydrochloric acid. Now this particular pepsin, okay, it will act on the protein present in the food. Let us say that you have taken some of nuts, groundnut you have eaten, or you have taken some grains, or you have eaten egg, anything, any protein food if you have taken, that will be digested first by pepsin. And what are the products of the pepsin digestion? You are going to see some polypeptides, peptones and proteoses, they are all of chains of amino acids. Different length of amino acid chain will be produced by breaking a long protein molecule which is differently folded. So that means by the end of the digestion in the stomach, now that partially digested food will have some polypeptides, peptones and proteoses. Okay. And because of this acidic condition of the gastric juice, pepsin was able to digest the protein. Now in the food there will be smaller portions of the or smaller chains of the proteins which are not much complex and easier for digestion also. Next, what happens, this food, okay, together with other components which are partially digested will be then thoroughly mixed and sent to the next region of the alimentary canal, that is the duodenum. Okay, duodenum is the first part of the small intestine. Now here what happens, this duodenum receives juices from other organs also, like it receives bile juice from liver, it receives pancreatic juice from pancreas, and pancreatic juice contains different types of enzymes. Okay, not only that, intestine has its own gland, it has its own secretory cells, they also secrete certain enzymes. Now let us see what are those different enzymes secreted by the pancreas. So pancreas secretes trypsin, chymotrypsin, carboxypeptidase A and B, and the proteins digesting enzymes involved from the succus entericus are dipeptidases, tripeptidases and aminopeptidases. Most of these are again in the inactive form. Okay, just I forgot to show you. So here this is the intestinal gland diagram what we have here, and there are different types of cells that are involved in digestion and absorption. Okay, I am concentrating only on these type of cells called as Paneth cells. Those are basically involved in secreting the enzymes in the intestinal gland. Okay, yeah. Next, moving on to the digestion of protein. So when we are talking about the enzymes, there are actually two types of enzymes. Okay, they are, one is endopeptidases, second one is exopeptidases. Peptidases refers to the enzymes that digest the peptide bond. A peptide bond is the bond present in a protein. Amino acids are joined together by a bond called as peptide bond. Any enzyme that breaks this bond is called as peptidase enzyme. Okay, and under those peptidase enzymes there are two types. One is endopeptidase, other one is exopeptidase. Now endopeptidases, they break or they cleave the bond within the chain. So now here you see, okay, as you can see here, this one, endopeptidase. Where it cleaves within the chain. This is the long chain, and within this long chain the breakage occurs. So here the breakage takes place. Within the chain the breakage takes place. Whereas exopeptidases, as the name itself says, exo, outer. So they usually cleave the terminal bonds. You see here, okay, this bond, the peptide bond that is present will be broken by the exopeptidases. So, there are endopeptidases and exopeptidases. Endopeptidases break the bond within the chain and exopeptidases break the bond at the terminal region of the chain. Okay. So, the enzymes that are involved in protein digestion are of two class. One is exopeptidase, the other one is endopeptidase. Okay. And now how exactly the digestion takes place, I think we can discuss in our next class. Thank you.